Hey, Evan.
How's it going?
Okay. He's back.
I'm back.
How you feeling?
Doing good.
How sick were you?
Luckily I didn't get sick. I tested negative. So I actually.
You tested negative?
Yeah.
Okay, so it was just, you were potentially exposed. This was just for safety's sake. Welcome back. When would you like to take the exam?
Good question.
I'll tell you what. Look at your schedule, get back to me. Can you take it by this week?
Yeah.
Okay. I'll just tell you right now, tomorrow works great for me. So if that works, if that doesn't work, Friday. Or if you want, I can give a copy to Mrs. Schaefer. You can go see Miss Schaefer, take it whenever you're ready.
I should be. 3:20 tomorrow's my last lessons.
Okay. Then you can come see me, or you can go see Miss, well, Kathleen might be going home by then. So why don't you just come see me? That way we don't. So, okay, terrific. Well, I did the Zoom thing all for nothing, but that's okay. I talked to Mrs. Schaefer about the temperature in this room. We're gonna see about getting the temperature turned up because the entire north side of the building up here is that way. Kelsey, did you get the email?
Yes,
You
thank
did,
so
yeah.
much.
From what I could tell, it went through.
Yeah, it went through, got received.
So excellent. Okay. I wrote you a humdinger, so they had better let you in.
Thank you, I appreciate it.
Absolutely. Absolutely. Well, if you have sweaters on and
I didn't know what I was doing on my quiz.
Second question was easy. Third question, I had no idea.
Gary, I am sorry about that mix up. Can you get it done? Either
No,
end of the week, beginning of next week, something like that? Okay, that'd be great.
So you did 10 instead of 1,000. That's tough.
I was so mad, I literally knew as soon as I turned it in, I was like, yeah, that was wrong.
Yeah, no, I got the first one right, second one
You
right.
already put your quiz?
No, but I know which ones I got.
Okay, gang, it's time to start. So I put a Zoom together because I thought Randy wouldn't be here. No, Randy shows up to class, so he tested negative. He's back. Welcome back, Randy. So the recording will actually be the Zoom recording. So I pulled out my microphone to see if it works, and it doesn't work very well. So I might have to get a new one. Some of those, the distortion on them is awful. When you record, you hear this. It's ridiculous. So the problem with this is when I walk away, you get a Doppler effect. So I can't really carry this around with me. That would be really dumb, but we'll do the best we can. All right, let's have a word of prayer and then we'll begin. Lord, thank you for your grace that gives us this gorgeous weather, your grace that gives us a chance to go to school here at this beautiful place. Your grace, Lord, so undeserved. And we thank you for coming from chapel. We pray now, Lord, that the message would resonate in our heads throughout the day, throughout the week, yea, even for the rest of our lives. Thank you for this time to learn about the role that what seems to us chance plays in the way living organisms change and adapt and have their population structured. In Jesus' name, amen. Now, it dawned on me that there were some things that I hadn't defined. So let me define them for you because I keep using these terms. It'd be better if I define them so you knew what in the world I was talking about. So there's this number that keeps coming up on slides and it goes like this. Have you seen that designation before in population biology or genetics, what have you? So that refers to the number of breeding individuals in a population. The number of breeding individuals in a population. So the number of ratio. Then we have this other term, and it's the term that's used up there, and I didn't really define it much, I kind of flew right by it. And with a subscript that says E, that is the effective breeding population. Effective breeding population. What's the difference? N is the number of possible breeding individuals. Effective breeding population, those are the ones who actually breed. So what if, so for example, turtles. We all love turtles, right? Baby turtles. Mom comes, dig, 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 lays her egg. Very, 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 she legs it back into the sea. The baby hatchlings, they come at a time, the eggs hatch, out they come, and they waddle their way to the ocean. But predators know that it's about time, and so the predators start to flock about usually birds. So usually the turtles will lay far more eggs than they need to sustain the population because they know the die-off is going to be pretty high early on. So the number of individuals that hatch and are anatomically normal and get up through the sand, that's your number of breeding individuals. Are all of them folks going to breed? No. Some of them are going to become seagull food. So that's the difference between your effective breeding populations, ones that actually survive, grow up to sexual maturity and breed, and N, the number of, I think maybe a better way to say that would be the number of potential breeding individuals. Number of potential breeding individuals. And so you have this ratio in conservation biology where you're looking at the effective number of breeding individuals divided by the number of potential breeding individuals. Does that make that clear? Is that better? It seems to me I didn't explain that. I just assumed you guys had heard that before, and that might not necessarily be the case. And I get on evals, I get dinged for that all the time. You just assume that we heard this stuff. We heard X, Y, and Z when we didn't. And I've also discovered that students don't always, because they're writing so quickly, have the awareness of mind, actually raise their hand and ask a question, because they're busy writing things down so quickly. So I define that for you, hopefully that makes that clear. Now, if that number is close to 1, what does that mean for the population? They're doing really well. They don't have many predators. There's not a lot of disease. The other thing about their doing very well would be they don't have a lot of lethal mutations that are going to be homozygous that cause early die-off or something like that. So if you have organisms where this ratio, so this is just theoretically with 65 different animal populations. If we look at this particular number here, we have conservation concern, stability, exploited, so on and so forth. So the red over here, look at that ratio there. What does this tell you? This tells you that you're looking at about 1 out of 20 individuals are going to breed. Now in the wild, that might very well be the case. So what do? You have lots and lots and lots and lots of babies. So for example, many of the organisms that shed their gametes into the ocean will overproduce gametes. They overproduce gametes because they know that most of those individuals that are formed from the fertilization of those eggs are going to become planktonic and they're going to be eaten by filter feeders. not a good day. But a 1 out of 20, that's an estimate that frankly is pretty optimistic for those kinds of critters. Whereas over here, if you're at, close to 1, your conservation concern is such that your conservation concern essentially, if your individuals get down to 26, then you start to have a concern. Otherwise, if your population is 33, then that's really not that big a deal. But as we get over here, now we start to see certain types of concern. So it's going to depend on how many individuals that you produce per offspring. Birds of prey, for example. How many babies do mating pairs tend to have for birds of prey? Not very many. Think about it. You have to hunt for them, and you have to hunt for yourself. You're expending a lot of calories just hunting. So to feed another mouth, that's a big energetic investment. That's why these birds of prey will tend to have only 1 offspring. Now, when this becomes the case, now you start to have some populations that essentially are stable, but others that can become problematic, depending upon essentially the number that you need essentially to be stable. So that's essentially what this is showing. Now, another thing I wanted to actually talk about that I didn't really talk about. There. This number here, 1 divided by 2 times the number of potential breeding individuals. When we talk about the neutral theory, so the neutral theory, remember, it's not that the neutral theory denies the existence of natural selection. It's that genetic drift is the greater contribution to change within a population. So the probability that a mutation will be fixed, that a mutant allele will be fixed within that population, when we talk about genetic drift, is 1/2 times n. You can see as the population gets bigger, that probability starts to go down. As the population gets smaller, that probability now starts to go up. That is how you calculate the probability of an allele being either fixed or lost. And we looked at examples of this. We looked at the Bury experiment in Drosophila and the theoretical ones. I don't want to read to the right act on that anymore, but we saw that this works essentially. All right, so back to the neutral theory. So Kimura's neutral theory, which is pretty much the best articulation of the neutral theory, it holds that although a small minority of mutations in DNA are advantageous and will be fixed by natural selection, the majority of those mutations will be disadvantageous and will be eliminated by something called purifying selection. Purifying selection. What's purifying selection? We're going to talk about selection next, but purifying selection essentially says that mutations that cause deformity, early death, infertility, things like that will not be carried in the population because their tendency to be passed to the next generation is low because of the infertility, inviability, or what have you of those particular types of mutations. The majority of mutations, therefore, will be neutral. What do we mean by neutral? They are not selected for or selected against by natural selection. They're neither selected for nor selected against by natural selection. That's what we mean by neutral mutations. So the majority of mutations, according to Kimura's theory, will be neutral. Will be neutral. So most genetic variation at the molecular level is neutral. It lacks adaptive significance. Did Kimura dream this up essentially after a night of bad pizza? No. Kimura came to this by examining amino acid sequences from various lines of vertebrates. When he compared the amino acid substitution rate between different lines of vertebrates, and when he looked at essentially the time in the fossil record when those vertebrate lines separated, he discovered that the rate of amino acid substitution between different vertebrate lines was constant. That's not what you'd predict if natural selection is the main driving force in those substitutions, in those amino acid substitutions. That told Kimura it was probably genetic drift that was the larger contribution to these mutations, not natural selection. Excuse me, the largest contribution to the fixation of these mutations in the population and not natural selection. All right, so it's this that leads us to the so-called molecular clock. When you get into the neutral theory, it gets very mathematical and very heavy, very fast. I don't want to read you the riot act on the neutral theory. I just want to give you a flavor for what it says. So I've got pages and pages of notes here with lots of equations. We're going to skip all that. Let's talk about tests, essentially, for the neutral theory. For the neutral theory. So, and this picture here is meant to illustrate essentially how this works in population. So if this is time, this essentially is the tendency for a allele essentially to either be fixed or be lost or something like that. So the time required for fixation within a population is longer in larger populations. We expect that, given that the probability of fixation is 1 over, that's probability of fixation. And folks, probability of fixation and the probability of loss are the same in the neutral theory. Probability of fixation of allele and the probability of loss is the same. You'd expect that, wouldn't you? That's hard to see. Probability of fixation. And being the potential number of breeding individuals in that population. Smaller populations, therefore, you'll have fixation much more easily. Others will be lost, as you can see here, others will get fixed. Fixation is faster for smaller populations, takes a much longer time for larger populations. Okay, stop right there. Make sure you're with me. Now, here is an experiment that was done by Kirby and associates. And what these folks did was they looked at the changes in the nucleic acid sequence of mitochondrial DNA between different mammalian species. Now, if the substitution rate is just straightforward, you'd expect a linear curve, a linear sort of distribution like this. Instead, you see this. What's going on? Why are you seeing this? Kimura looked at this data and he said, I think I know what's going on. And he had to adjust his equations for this, and this experiment essentially told him why. Think about this. Oh, that's Councilman these racquetballs. Almost to destroy things. Let's see how rather NBM. Think about the genetic code. So you have 6 different. Well, let's just do this. Okay. Let's say you have a nucleotide sequence, and you've got something like that. This A. So this A can be changed to a T. Then it can be changed to a C. Maybe it can be changed to a G. But what if it gets changed back to an A? Or what if it can also be changed back to an A like that as well? This is going to skew your differences between DNA sequences, isn't it? Things are actually changing back to what they were like rather than becoming more different. Because you've only got 4 bases. So as you change from one base to another, there's a possibility you could change it back into what it was in the ancestral condition. That's why you don't get this linear. It's linear at first, and then as time goes on, it starts to skew this way. And Kirby's experiment essentially showed that it's just not this nice little linear thing, because you've only got 4 bases. You potentially, one of the changes could be to change back into the ancestral condition. That's why it's going to do this. So this is from the Kirby paper, and this is mitochondrial DNA from 2 human individuals. If you add more human individuals, that guy up at Halifax University, Mike Gray, has extended this to 843 or something human individuals, goats, sheep, and then you can use other hoofed animals, cows, pigs, horses, camels, what have you. Pairs of primate species, you can then extend those to other non-human primates as well. Pairs of rodents, rodent primate pairing, so on and so forth, blah, blah, blah. You can send those to further and include other mammalian species in here as well. This holds. This curve holds. It looks like that same shape. So even if you put more individuals in here, you're still going to curve like that. Because, well, this is what you might expect if it was just changing. Those changes can sometimes go back to the ancestral condition. That's why you get this. So Kimura now has adjusted his equations, and the equations that he has used give a better fit for these kinds of data. So from this, a guy named Hartle, Bob Hartle, did a theoretical analysis and said, if the neutral theory, if we take genetic drift to be essentially the main driving force within a population, we would expect, given if the effective population increases, here's what we would expect essentially for the heterozygosity of the population as the population gets bigger. Remember, it takes longer essentially to lose or to fix alleles in a larger population. In smaller populations, you're going to tend to lose certain alleles, but as Kimura pointed out, you're going to be introducing new alleles through new mutations over time. Some of those will be fixed, some of those will be lost, so there will be a steady state within the population, a steady state. What is, therefore, that level of heterozygosity that you would expect within populations? Hartle and Clark. Hartle and Clark essentially did a set of theoretical calculations to show essentially as the populations get bigger within a certain size, this, as you can see, is going to asymptote to right about here. This will be your equilibrium heterozygosity here. So you would expect relatively high levels of heterozygosity in large populations if, in fact, the neutral theory is the main driving force in populations. Now, remember I told you about that finding of Lewontin and Hubby? Lewontin and Hubby in their electrophoretic analysis of different, it was mainly insect populations, but then Gottlieb's follow-up work on plants confirmed Lewontin and Hubby's work that in fact polymorphism was the rule. Heterozygosity levels, in fact, in natural populations are very high. This predicts that in large populations, your heterozygosity is going to be very high. Does that match with what was seen by Lewontin and Hubby and Dojemski and Gottlieb, all those other people? Yes, it does. That suggests, at least in big populations, genetic drift is playing a much larger role than people originally thought. Genetic drift is probably playing a much bigger role than people originally thought. Keep up with the Zoom here. All right, so then this is another set of experiments that examined nuclear genes. So this other study looked at mitochondrial DNA. These are now looking at nuclear genes between humans and then 9 other vertebrates plotted against the estimated time from the last common ancestor. And we have to introduce some other terminology here. So if genetic drift is one of the main drivers, it seems, of populations, what role does selection play? And can you detect selection in a population? The answer is, it seems to be is yes. And here's how you do it. Think of the genetic code. You think of the genetic code, some of those codons code for more than one amino acid. Others code for but a few amino acids. If natural selection is keeping amino acids at certain positions within the protein, then you would expect the changes that occur within that codon to be synonymous. What does that mean? Synonymous. Synonymous. What's a synonymous codon? Yes, exactly. Kimberly is absolutely right. Code for the same amino acids. So synonymous change does not change the amino acid coded for in that protein. Non-synonymous changes non-synonymous nucleotide changes change the amino acid sequence. So if you take a look at the ratio between synonymous and non-synonymous changes in a protein coding region, you should be able to detect if natural selection is occurring. Natural selection is not occurring. The ratio of synonymous to non-synonymous changes should be about the same. If selection is occurring, then the number of non-synonymous changes, the frequency of non-synonymous changes should be greater than the frequency. No, the frequency of synonymous changes should be higher than the frequency of non-synonymous changes because you're keeping the amino acid sequence the same. Let me say that again. If selection is present, the frequency of synonymous changes should be greater than the frequency of non-synonymous changes. If selection is not present, the frequency of those two changes should be about the same. You see why that makes sense. If genetic drift is playing a role, a greater role in the evolution of that protein coding region, you should be able to change any old place, and it shouldn't matter if you change amino acid sequence. So your non-synonymous, which will change amino acid sequence, and your synonymous changes, the frequency of those two changes should be about the same. That's if there's no selection present. If there is selection present, then your synonymous changes should exceed, the rate of your synonymous changes should exceed the rate of your non-synonymous changes. That's what these funky numbers here, D, S, D sub A, D sub N, that's what they mean. That's what they mean. So D sub S, that is the rate of synonymous changes. The rate of synonymous changes. D sub N, those are your rate of non-synonymous changes. Non-synonymous changes. And then D sub A is a analogous changes. That is, you get a change to an amino acid that is similar, but not completely different. So some amino acids have similarities chemically, and it turns out in the genetic code, you can change, for example, a serine. Serine has a hydroxyl group in it, so does threonine. And it turns out if you change one of the bases in the serine codon, you can change it to a different amino acid, but you'll change it to a threonine. That's a so-called conservative change. So whereas threonine is a little bulkier than serine, but chemically it's the same as serine. All right, so if you look at these 4,198 nuclear genes, looking at human and other 9 vertebrates, and you're plotting them essentially per unit time of divergence. So what do you see here? These points fall on a line. Does that convince you that's a line? Convinces me. So this essentially is some of these critters that's essentially you're looking at. There's groups of genes essentially that show essentially that the degree of non-synonymous changes, that the frequency of these non-synonymous changes essentially is about equal to the number of these synonymous changes. This tells you that these particular genes, this cluster of genes, some of these 4,198 genes, looks like genetic drift is the main thing changing them. Another group, there's a little more constraint on them, it seems. You can change, but you got to change them to things that are a bit chemically similar. Then there's this other group of genes here. Those look like there's a good deal of constraint on them. These only are going to tolerate synonymous changes. So this tells you that some genes have respectable constraint on them. Selection's playing an important role in these genes. For example, you guys heard of histones? What do histones do?
Say, are they used in the production of histamines?
No. Histamines are small molecules derived from amino acid histidine. You decarboxylate becomes histamine. Histones are used to package DNA. So you take a histone protein and that's a histone protein. And you take 8 of these and you make a spool and you wind the DNA around that histone spool, and then you package your DNA that way. It helps make chromatin. Histones between different species are very, very, very highly conserved. You take a histone from a calf and a histone from a pea plant, there's 2 amino acid differences between them. So very similar, very similar. These are under strict constraint. These would be in this particular group here. These are under high constraint. Yeah.
So what happens to the stone if you change a lot of amino acids?
Then it doesn't work anymore. You don't make chromatin, you don't package your DNA properly. The cell isn't really able to function because the cell has a lot of DNA in it. You have to be able to package it, excuse me, into a proper chromatin. Then you have to be able to regulate the winding and unwinding of that. Also, you have to be able to cinch chromosomes down into proper chromatin, heterochromatin, in order to make metaphase chromosomes so that they can be distributed during mitosis.
So is that a known disease?
No, because people with mutations in histones don't tend to live. So we don't have individuals to study with mutations in histones. They tend to go back up, so to speak. So that's an example of a protein essentially that's in this group here. Other proteins, so essentially it's going to depend which protein that you're looking at. So now the plot gets a little thicker. So while we might have had things like this, and we're starting to see things like this, it looks like the neutral theory is the way things work. Not so fast as, who's that guy in the college football, ESPN, Jeremy, not so fast. I can't remember his name. He's very entertaining. Anyway, whatever his name is. He does the picks and he puts the mascot's head on when he makes them. Yeah. I can't remember his name. Yeah, not so fast. You know the guy I'm talking about. Okay, I can't remember his name either. But if you watch college football today, he's always on there, and he's kind of a riot. If I have half his energy when I reach his age, I'll be doing pretty well. He apparently, when he's younger, was quite the football player. Yeah, he's like in the College Hall of Fame or something. So he was no slough when he was younger. So not all genes essentially show this prediction by Kimura that natural select that genetic drift is the main thing governing their change. Selection seems to play a respectable role. Now, here's the hitch. For the majority of genes, is that the case? That's the part that's still being debated. Because it seems like for most genes, it's a mixture of selection and drift. So which is the bigger force? That's the question that's still being debated. I actually looked at some textbooks that were 20 years old. It was a debate 20 years ago. And then you look at the textbooks and even the papers 20 years later, it's still being debated. It still hasn't been resolved. Now, here's where the neutral theory really does very well. When you look at the rate of change amongst different types of
regions of genes. You notice that this is supposed to be, so the URL flanking, so the untranslated region, the 5' flanking region, 5' untranslated flanking region, your non-degenerate sites, twofold degenerate sites, fourfold degenerate sites, introns, your 3' untranslated region, your 3' flanking region, and then your pseudogenes. Who in this class doesn't know what a pseudogene is? 1, 2, 3, 4, 5, 6. Okay, let's talk about a pseudogene. A pseudogene represents a gene that either stopped being used, it stopped being expressed, so it now just essentially accumulates mutations. You also see this in genes that are duplicated. The duplicated version isn't used. The organism doesn't need it, so it just kind of starts to accumulate mutations kind of at random. Good example in our bodies, we have the gulonolactate oxidase gene, which is used to make vitamin C. We get vitamin C from our diet. We don't make vitamin C, so our gulonolactone oxidase gene, the GLO gene, has accumulated mutations. That gene in our bodies, our genomes, is a pseudogene. So if you look at the rate essentially of substitutions per nucleotide site, you'll notice one's essentially where you don't have degenerate sites. So like methionine. Methionine is encoded by one codon, AUG. You can see the substitution rate is less than 1 per site, very low. When you start to get to twofold, fourfold sites, now your rate of substitution goes up. When you get to regions outside genes, where you don't have coding, essentially, now essentially your rates of substitution go up. When you get into introns, they go up, and the highest rate of substitution is in pseudogenes, because there's no natural selection essentially restricting the substitution rate in the pseudogenes. The best explanation of this is the neutral theory. The best explanation of this is the neutral theory. With introns, you have a little bit of constraint because you have some sequences that are needed for RNA splicing. With pseudogenes, there ain't nothing holding you back from changing that thing. There's some regulatory sequences in some of these, that's why there's some constraint here. But essentially, the more degenerate you get, the more substitutions you get, and in pseudogenes, you get the highest. The neutral theory does the best job of explaining these data. This is a case, as you can see here in the title. This data comes from humans and rodents. This is from a paper that came from 2003. This has been augmented by work done by the Harvard guy, his name will come to me. I can't remember his name now. And this holds. This same sort of graph essentially holds if you expand the data set. The neutral theory does a very good job of explaining this. And so if you now look at different plants and you look at essentially nucleotide substitution rates between organisms, when you do trees like this, longer sorts of lines represent that you have higher rates of nucleotide substitution in these particular lineages and what have you. So here is your spore-bearing plants. So Equisetum, that's your horsetails, your ferns, angiosperms, those are your flowering plants, and then ginkgo, these are your cone-bearing plants, gymnosperms, things like that. Ephedra and Lavixia and things like that. These are pinus, of course, pine trees, other things like that. Why do you get differing rates, essentially, of nucleotide substitution in some of these different organisms? The reason seems to be, in some cases, which organism is going to have the change in the nucleotide sequence? It's going to be the progeny. What if your one organism is longer lived than another one? Pine trees. How long do pine trees live? House I grew up in, there was a pine tree outside my bedroom window. It was there when my mom and dad bought the house. Last time I visited my hometown, we drove by the house, that pine tree's still there. That pine tree probably is over 100 years old. Is that uncommon for pine trees or common? Pine trees have a pretty long life cycle, don't they? So you can see that line is a little bit short. What about these pups over here? Ephedra, I don't know if any of you are familiar with ephedra. Ephedra is a little weedy thing, and it doesn't live very long. So it generates a lot of progeny. That's why you get these different rates of substitution in different lineages. Often because organisms live for different periods of time, that means the amount of progeny they're going to leave over a period of time will differ. The more progeny you leave, the more opportunity there is for changes in nucleotide sequences. The longer your lifetime, the less progeny you tend to leave, the less opportunities there are for changes in rates. That's why you get these different lengths of lines. Now, can this explain all the disparities in nucleotide substitutions? The answer seems to be no. Let me give you an example. And this comes from John Gillespie. Where is it? So if you look at the, oh, here it is. If you look at the rates of base pair substitution within two mammalian orders, primates and rodentia. So primates, that's humans, non-human primates. Rodentia, obviously rats, mice, things we love to hate, what have you. Those are two mammalian lineages, and rodents branched off the mammalian stem earlier than primates, but because the fossil record of rodents is pretty good within the parts of the Cenozoic, you have a pretty good handle on when modern rats and mice essentially arose. So if you look at the substitution rate between, say, humans and chimpanzees, and then rats versus mice, the substitution rate is much higher in rats in the rodent line than it is in the primate line. Can that be wholly explained by the longer lifespan of humans versus the lifespan of mice and rats? No, it can't. So that has not been completely resolved. So just so you know, sometimes when you see these trees, you see longer lines. That longer line means you have higher rates of nucleotide substitution. That may be due to their shorter lifespan, but it may just be in that lineage you have higher nucleotide substitution rates. Keep that in mind. Yes?
I was just wondering why that would be, because even like with plants that live longer at the time, they're reproducing in a year.
It might depend on the lifestyle of the plant. So if the lifestyle of the plant is such that dispersal is very wide and the plant is adapted or adapts easily to many different environments, it could be that a higher mutation rate is actually to its benefit, especially if it leaves a lot of progeny. And it's got progeny to burn with a higher mutation rate. it's clear that the mutation rate is also under natural selection. That's something we'll look at when we talk about selection. It could be in some plants, especially fast-growing plants. That might be something actually that the plant wants to do so that its ability to adapt is greater. It's a trade-off. There's less stability in the genome, but that means that the plant is more adaptable. That might be. That's one explanation for certain types of fast-growing weeds, essentially. Some of them actually have higher mutation rates than some of your flowering plants that are more stable, but are more restricted to certain environments. All right, so here's our old buddy, the pocket gopher. And you remember from the pocket gopher does not tend to walk between its different locales. So here's California, Nevada, Arizona, Utah, Colorado, New Mexico, so on and so forth, and then down into, this would be Texas over here, this would be the Chihuahua Desert down here, and what have you. And these are frequencies of certain alleles of various genes, of two genes. So ADH, alcohol dehydrogenase, LDH, lactate dehydrogenase. Lactate dehydrogenase will convert pyruvate into lactate. So it's used in muscles. So if the animal, for example, this is a digger. If the animal is digging a lot and the animal starts to show fatigue, it'll start to make lactate. These animals can dig at a high rate, actually for a long time, because the lactate tends to be circulated quite readily through the animal's body, and therefore it doesn't tire. The muscles don't cramp very easily. And it turns out that different alleles are correlated with different hardnesses of soil. And certain alleles tend to help the animals dig through harder soil. And in some of these desert areas, you have this stuff called hardpan. Maybe you tried to dig through hardpan? Yeah, so hardpan is a result of clay. You get clay soils essentially that dehydrate, and you get these layered clays, and the soil does this, becomes as hard as concrete. When I was landscaping oh gosh, it was my junior year, I think, going into my junior year in high school. I worked for a landscaping outfit. We were putting in a pool. Got out of our truck, went to the backyard, took our shovel, broke the shovel, hard pan. So we got out this thing, which in landscaping we call a sharpshooter. And we sat there and we hacked and hacked and hacked. And then we went out and got the pickaxes. We hacked at that thing all day in the 104 California heat. And after whacking at this thing all day, we'd gotten, I think, about that deep. So we called the demolition guy. He drilled a bunch of holes and he put some dynamite in. And he dynamited it. And then the next day we came back and shoveled away a lot of the debris. And the next day we were about that far, 2 days. Then we had to have him call, we called him again, and had to drill some more holes and do it. And then we had to dig a hole for a tree, to put a tree in. And we broke a sharpshooter doing that one. So we called him again and put some dynamite in. Yeah, anyway, that's my story of hardpan. So these guys are trying to dig through hardpan. That tells you essentially the stamina some of these guys have. And hardpan, you'll see hardpan in some desert environments. Anyway, alcohol dehydrogenase and lactate dehydrogenase, here's different alleles of these. So there's 4 alleles here, there's 3 alleles. And then as you look essentially at these populations here, these populations again are pretty isolated. Tell me what you see.
Doesn't seem to be anywhere else.
Yeah, these particular populations, they vary pretty greatly, even though they're nearby. You getting much gene flow between these? What does gene flow tend to do to allelic frequencies? Tends to make them the same. You getting much gene flow between these? No. This is genetic drift. This is exactly what you'd expect to see if genetic drift is playing a bigger role in the molding of these populations. Because these guys, they don't move very much. They're established essentially by largely something called sweepstakes dispersal. And these are populations now that are very isolated from each other. This is largely due to genetic drift. Okay, I'm going to skip that and move on. All right, so another consequence of genetic drift is this thing we see called bottlenecks. Bottlenecks. And if you get a small representative from a population establishing another population on an island, on another place, you have some sort of geographic barrier, you then get isolation, something like that, or some outfit leaves and goes someplace else, essentially, you get this thing called a founder effect. And the founder effect essentially represents a induced bottleneck. So this, a good example of a bottleneck, comes from the human population. It's clear, and we'll take a look at this when we look at ancient life, but it's clear that the human species originated in Africa. Reason being is that if you look anywhere on Earth, the largest amounts of diversity, genetically speaking, are in Africa. Furthermore, all the other genetic variants that you find elsewhere are easily traceable back to Africa. So it's pretty clear that the human species originated in Africa. So from this group that left Africa, some of that group went north and became essentially Europeans. So these individuals diverged somewhere about 23,000 years ago or so. This group that left Africa seems to have left, the European lineage left about 51,000 years ago. And then the divergence where they became genetically distinct was 23,000 years ago. And about that same time, you had another group migrating on a different route, essentially. Instead of going north, they went directly west and then south. They essentially became the line that gave rise to the Asian, the so-called Yellow River, the Yellow River civilization. And that's your Chinese and Japanese and other populations as well. These are two different bottlenecks, but then diverged along different lines as well. This is an example of bottlenecks. And you can see bottlenecks, but again, from this bottleneck, because these two populations were able to spread and adapt to other microenvironments, they then became very diverse, but their diversity, you can see, is a derivative of this greater diversity here. That's why it's pretty clear these individuals came from this larger diverse population in Africa. And this is merely essentially showing linkage disequilibrium between certain genetic markers in human populations. In order to have linkage disequilibrium, you have to have natural selection. And as we go away from Africa, your level of linkage disequilibrium, this is looking at specific loci within a 10 kilobase range, notice it falls upon a straight line. That tells you that selection is playing a role, and it's playing a role in more populations than it is in others. So here's the population, the Bantu. Did they leave Africa? No. Here's the ones who left Africa. They have higher rates of linkage disequilibrium. Your degree of linkage disequilibrium increases the further you get from East Africa. That tells you that you had selection essentially working on those populations as they left Africa. Okay. Any questions about the neutral theory, about genetic drift, anything like that? Because the next thing we're going to talk about will be selection. I just have time. You know what? Forget that. We'll pick up with selection on Friday. So we do have a quiz Friday. It'll just be over the neutral theory and over genetic drift. I will keep it on the down easy. I won't get too deep in the weeds with the math and everything. Do you guys think you have your heads around the neutral theory? At least a basic bare bones. If I asked you to explain the neutral theory to your roommate who's an art major, you could do it. Gary's sitting over there saying, art major, come on, please. If I asked you to explain it to another friend of yours who's a major, bio major, you could do it. Okay, we'll practice. Practice, Gary, practice. All right, so that will be your quiz. We'll hit selection then on Friday. We'll take a look at how selection, we'll see selection is not one thing. There's different types of selection. We'll take a look at how they skew Hardy-Weinberg ratios. And then we'll look at some examples of them in the wild. Then we'll see how these can actually team up to actually cause populations become so different that they can no longer breed with each other. And that will guide us into our discussion of speciation. Okay, we'll see you on Friday.